Hi, we made it pretty far in microbiology. In this presentation, we'll describe the gram staining process to distinguish between gram-positive and gram-negative cells. In the past couple weeks, when we've been looking at bacteria under the microscope, we've looked at cells that had been simply stained, or just stained with one dye, like crystal violet or methylene blue, so that they showed up on the microscope slide. Differential staining is a different type of technique that uses series of different stains to allow different morphological features in bacteria to look different, much like differential media caused bacteria to look different based on a particular biochemical property. In gram staining, we're using a differential staining technique that helps us distinguish between two different morphologies of bacterial cell walls. As examples, on the left-hand side of our screen, we can see Staphylococcus aureus cells that were stained purple because they are gram-positive, which is different than the way E. coli, a gram-negative bacterium, stains, and they appear pink after this gram staining process. The reason that the gram stain as a differential stain works is because different classes of bacteria have different cell wall morphologies. When we describe bacterial cell walls, we tend to include the cell membrane and whatever cell wall material exists outside the cytoplasm of the cell. So on the left-hand side here, we have an example of a gram-positive bacterial cell that has a cytoplasmic membrane and then a thick peptidoglycan cell wall on the outside of the cell. So peptidoglycan is a polysaccharide matrix. We could think of it similarly to a cellulose cell wall in a plant, but it's cross-linked with small polypeptides, and this is a very rigid structure that gives bacterial cells their shape. So let's remember that gram-positive cells have a cytoplasmic membrane and a very thick peptidoglycan cell wall on the outside. And this is to be contrasted with the gram-negative cells, which instead have an inner membrane, like the gram-positives do, but then they have a very thin peptidoglycan cell wall depicted here, followed by a second membrane, which is the outer membrane. And the differential staining nature comes mostly because of the differences in the size of that peptidoglycan cell wall. Gram-positives are very, very thick, and then the gram-negatives are very, very thin. And when we look at a gram stain that has mixed species, we can see that the gram-negatives with their thin peptidoglycan cell wall stain pink, and the gram-positives, which are represented by the Staphylococcus here, stain purple, like we saw on the slide immediately prior. So how do we go about producing one of these gram stains? It all starts with a smear, and you are all experts at this, so I'll go through this pretty quickly. You will have placed a sample of bacteria, either from a liquid culture or from a petri dish, into a small drop of water on a microscope slide. Spread it out very, very well so that it dries into a single layer, and then you've heat fixed that on the slide with a Bunsen burner so that the outer proteins on the outer membrane denatured and stuck to the glass. That holds the bacteria in place. Now we can follow up with some staining, much like we did in the simple staining last week. Simple staining was a two-step process. We added a stain, let it sit for a while, and we rinsed it off with distilled water and then looked at the cells under the microscope. The gram stain is an 8-series process. We start off by staining our cells on our microscope slide with a primary stain, which is called crystal violet. And this stains every single one of our bacterial cells dark purple. After they've been stained for 30 seconds to a minute, we would rinse all the excess stain off of the slide with a little bit of distilled water. And likely, in the positions where we had bacteria, we would see that there were nice dark purple patches because all of the bacteria were stained. Next, we're going to add a chemical called Gram's iodine, which serves as a mordant. So if you think about a brick wall, all of the bricks are stacked up, and separating and holding all of the bricks in place is a white compound called mordant. So the mordant serves the purpose of gluing together all of the bricks in a brick wall. In a gram stain, the mordant cross-links and it binds to multiple molecules of crystal violet. So by adding Gram's iodine to the cell wall of bacteria that already has a lot of crystal violet in it, it increases the size of those complexes and it sort of locks it in place so that the crystal violet remains stuck inside the cell wall. That's a very important point, and it's going to help us out in our next step. After adding the mordant or the iodine to our stained crystal violet cells, we'll rinse that back off again to get rid of all of the excess iodine. Now at this point, every cell, whether it's gram-positive or negative, is going to be stained purple, and the color will have shifted a little bit after being cross-linked to the iodine. The next step is the most critical step of the gram staining process, and that's to decolorize the cells. And to decolorize, we're going to use ethanol. And ethanol is going to dissolve the outer membrane in the gram-negative bacteria, and it's going to solubilize the crystal violet that's being held in the peptidoglycan cell wall. Gram-negatives have very little peptidoglycan, so it's pretty easy to wash away that crystal violet, even though it's been cross-linked with iodine. Gram-positive bacteria, on the other hand, have a very thick cell wall, so the iodine is not as efficient at drawing the crystal violet out. During this decolorizing step, all of the gram-negative bacteria will lose all of the pigmentation and staining that they've had, and they will become colorless again, as if they had never been stained. But the gram-positive cells will stay purple, because they're going to retain a little bit. If you were to look at those on a microscope slide at this point, you would not be able to see the gram-negatives, but the gram-positives would still be stained purple. If you want to be able to see those gram-negatives again, then they have to be counterstained, or we have to add a stain to those cells to make them show up, but a stain that's not going to be visible, it's not going to show up in the presence of crystal violet. So we use a chemical called safranin, not saffron. Saffron is a very expensive spice to make your rice yellow. We use safranin, which is pink, and then that stains all of the gram-negative bacteria pink, and it doesn't affect the gram-positives, which have already been stained purple. That makes it differential. Bacteria show up in two different colors based on their external cell wall morphology. Gram-positives with a thick peptidoglycan cell wall retain the purple crystal violet stain, even after they've been counterstained with a little bit of the pink safranin. E. coli that have a thin peptidoglycan cell wall will have had all of their crystal violet washed away with the ethanol, which makes them transparent, and then it makes them available to be recolored with the lighter pink colored safranin stain. This is a great technique because many pathogenic bacteria are either gram-positive or gram-negative, so a quick gram stain, which only takes a couple minutes, will help us break down and figure out which class of bacteria we might be looking at.